right. Welcome. So if you haven't turned in your documentary assignment, I'll take that now. I have received a few emails. So that's fine. Whatever's most convenient for you. Talk about this in just a few minutes. Things that influence endurance performance, because that's one half of exercise science is the aerobic side, the other half is the anaerobic side. So as you can see from our schedule, we're doing really well staying on track, which is kind of rare because we usually go off track. But next Tuesday is your first exam, chapters 1 and 2, and so there's a study guide posted, and I'll show you that. So study guide is here. This is not an answer key, so please utilize all your resources, PowerPoint, textbook chapter, assignments, and so on, but this is very helpful. Points of emphasis for the exam. So the exam is all short answer essays, so you really have to know the material. Able to explain things, explain the processes involved, where ATP comes from. There'll be heavy emphasis on carbohydrate and fat metabolism, nothing on protein metabolism or hormones. So I'm saving that for later in the semester. So there is content on protein metabolism and hormones right at the end of chapter 2. I don't want to go into that now, so I'm going to wait a little bit on that. So there should be enough content here though to keep you busy preparing for exam 1. So as you look those things over, Thursday would be a perfect time to come if you have questions, and then please ask, because if you have a question, chances are everybody else has the same question. So
if we study the study guide and then just the content around the questions on the study guide, we should be fine.
Yeah. Don't rely too much on the study guide though, but it is a great tool. Okay, so you can access that. You can see in that folder right there. It's available. So okay, so my goal for today is to hopefully finish chapter 2. Making sure that we're understanding the concepts along the way. I want to talk about the documentary though. So factors that affect endurance performance. I thought it was really interesting looking at different groups of athletes from different countries and assessing all kinds of factors that are important for endurance performance. So maybe we could list some of these factors that were studied in the documentary, as well as a few others that will be talked about in your textbook. So there's more than 5, but we could start there. So what were some of the things compared between groups of athletes?
Diet.
Yeah, okay, so nutrition. Did they find much of a difference there? Hi, Mike. So nothing there. What else did they look at? So that's a little subjective, but maybe exertion level. Was there an objective measure that they could?
We did like a lot of
Yeah. So lactate analysis, which the test that they did was probably looking at the anaerobic threshold, sometimes called the lactate threshold. So we have our three metabolic systems, right? And so if we're able to handle the exercise intensity using primarily the aerobic system, everything is great, but eventually exercise intensity can change where the aerobic system is unable to meet the demand. And so what do? If we can't meet the demand for ATP mostly with the aerobic system, what else have we got to rely on? What other energy systems do we have other than oxidative? Yeah, glycolytic and phosphagen. So what's different about the glycolytic and phosphagen versus the oxidative? It's anaerobic. So how long can we stay in anaerobic metabolism? Not very long. Okay, so there's a threshold between where we're primarily emphasizing oxidative to where we've crossed over into the area where we're using both oxidative and glycolytic. And so that threshold, that boundary, where we increasingly rely on glycolytic metabolism to meet the energy demand is called the anaerobic threshold. And so the higher your anaerobic threshold would mean the longer you're able to stay in oxidative metabolism, which means you can maintain a really fast pace because you haven't crossed that threshold. Once you cross that threshold, you're talking about a few minutes, not very long, until your muscles would fatigue where you'd have to lower the intensity. So the higher that threshold, the longer you can maintain a very, very high level pace, which is an advantage for endurance performance. So they didn't find any difference there. So what else did they look at? Yeah, wing. Okay. Yeah, so that makes sense. In terms of, how much ground you can cover with each stride is mechanically would be an advantage. They didn't find any difference there. What were they doing with the biopsy? What were they looking at there with taking a piece of the muscle tissue and then freezing it and then cutting it up into little pieces and then looking at it under a microscope? What were they looking at? Muscle fiber composition, right. So that's another factor, muscle fiber composition. So based on our discussion here, maybe in other classes, if you're an endurance athlete, what type of fibers are ideal?
Slow twitch.
Yeah, so slow twitch. So that's kind of it's an oversimplification because there's lots of categories and subcategories between slow twitch and fast twitch. But it's really easy just to think of things as relatively slow and relatively fast. Slow is only really slow compared with fast. Fast twitch, they reach peak tension in about 50 milliseconds. Slow twitch can reach peak tension in about 1/10 of a second. So fast twitch are twice as fast. So slow twitch fibers are very fatigue resistant. So what could be other characteristics? Looking inside a slow twitch fiber, what would you find versus a fast twitch fiber? Let me give you a hint. Where does oxidative metabolism take place mostly? Where does the oxygen go? Yes. So more mitochondria, larger mitochondria. What's the molecule that transports oxygen from the muscle cell membrane to the mitochondria? So we're looking at increased mitochondria, larger. mitochondria and increase, the question that I'm asking, and the response I'm looking for is myoglobin. Myoglobin has a very high affinity for oxygen. So as hemoglobin carries oxygen through the blood, myoglobin carries oxygen through a muscle cell. So fibers are also smaller. Slow twitch fibers are smaller, and that has to do with diffusion distance. So the oxygen doesn't have to travel as far to reach the mitochondria with a smaller slow twitch fiber.
Does that help in fatigue resistance?
I think so. Fast twitch fibers are larger in mass. So if you think about an athlete, if they have a lot of mass, that's not good for endurance because they have to carry that with them. So we want just enough mass to be functional, but not so much that it's detracting from efficiency. So that provides a great bridge into the next. What about efficiency? Was there a...
Is that what you're talking about? Sorry.
No, not yet, but we can go there. And then we need to do that because this is the one that was different. So how do we define VO2 max? So you can estimate it. Like in the 106 lab, you estimate it. In this lab, we measure it directly. So we have some nice equipment. You can get a direct measurement of it. Brady, you have some big shoes to fill there. He was in this lab last year and went 15 minutes on the Bruce protocol. So soccer athletes have wonderful endurance. And you think about that sport, they're running like 7 miles in a match. So VO2 max, how do we define it? The maximal amount of what? Oxygen you can consume. So that's the criterion measure of aerobic fitness is your oxygen consumption. So that varies. So someone with average fitness might be, 40 to 45 milliliters per kilogram per minute, whereas an elite endurance athlete might be up in the 80s milliliters per kilogram per minute. So I think we measured Brady around 65, which is like consistent with a good collegiate cross-country runner would be 60, maybe even low 70s. So I think we measured Ace and Logan at 67 and 68, somewhere around there, which is great. Okay, so getting down to the very end of the documentary, very simple. What did they find that was different? And it has a lot to do with this concept of efficiency. Yeah, leg, body.
Volume.
Leg, yeah, yes, good. Volume. Leg volume. So the size of the legs. So how would that relate to efficiency? So the way that we measure efficiency in exercise physiology is how much oxygen are you consuming for a given speed? So you have 2 athletes, a Kenyan athlete and a Danish athlete in this study, and both athletes are running along at, say, 10 miles per hour. And the one that's running along at 10 miles per hour and consuming less oxygen would be considered more efficient. So it's like a fuel-efficient car. You can compare 2 cars, and they can both be traveling at the same speed, but the one that's consuming less fuel is considered more efficient. So it's oxygen that we're talking about in terms of fuel efficiency. So being able to run along at a lower percentage of your VO2 max. And so that would relate to leg volume because every time you flex your hip and you go to take that next stride, you're carrying that mass. And so a heavier leg, even if it's just a little bit, means more oxygen consumption, more energy expenditure. So there's a direct relationship between how much oxygen we're consuming and how much energy we are expending. So it's something like for each liter of oxygen we consume, we expend 5 calories approximately. So a more efficient athlete might be expending less than 5 calories. So if you're talking about several miles, that can make a big difference. So you're not fatiguing as quickly and so on. Okay, so any questions on the documentary? I thought it was kind of interesting, and I would have expected to find more differences with anaerobic threshold, VO2 max, something, and then it just all came down to just anthropometrics. So having really small calves, essentially. So.
I thought genetics would play more on that.
Yeah, you know, and they really didn't get into that, but there are certainly genes that are connected with good endurance performance. So there's a lot of research going on there to identify certain genes. The problem with that is you identify one gene that is correlated with good endurance performance, but there's probably 1,000 other genes that interact with that single gene. And so it gets really complicated understanding how all those interactions take place. Companies have made lots of money trying to isolate certain genes that are connected with endurance versus strength and power and so on, but it really is not a single gene. It's probably a lot more complex than that. Okay, so we've been talking about metabolism, glucose, fatty acids, sometimes amino acids. What's the common intermediate? So where do all these fuels eventually form a common molecule in the mitochondria? So, yes, acetyl-CoA is our common intermediate. So regardless of the substrate, metabolism proceeds in exactly the same way once you have acetyl-CoA. So why is it that for one molecule of glucose from the blood, we ultimately make 30 ATP, and for one common fatty acid like palmitic acid, our net ATP is 106. Has to do with the amount of hydrogen in one molecule of glucose versus one fatty acid. Because eventually we're taking those hydrogens to where in the mitochondria? Where are we ultimately taking these hydrogens? So we have these taxis, NAD and FAD, that are stripping hydrogens from our substrate and shuttling these hydrogens to eventually the electron transport chain. So that's where we make the large majority of ATP. Anaerobically, we make very little ATP, but what's the trade-off? Anaerobic ATP production is, we don't get very much, but it's fast. Fast ATP production to meet high energy demand. Doesn't last very long. With aerobic metabolism, we make lots of ATP, but what's the disadvantage? It's very slow, so it takes time to completely, we could say, oxidize glucose or fatty acids or sometimes amino acids. So what does it mean to oxidize something? So ATP production, as we break down glucose, fatty acids, or amino acids, it involves a series of oxidation reactions. So oxidation means that we're adding oxygen, or in this case, we're taking away hydrogens and electrons. So as we go from glucose to pyruvate to acetyl-CoA, and then we're completely breaking down acetyl-CoA as part of the Krebs cycle, we're oxidizing that substrate. We're taking away hydrogen and the associated electrons. So that's what it means to oxidize something. And so the NAD and the FAD are shuttles in that process. So they take away the hydrogens and electrons and shuttle them to a spot where we can make the majority of ATP. Now, there's a key difference. This is a good test question. That's why it's highlighted in red, between skeletal muscle and cardiac muscle. So in cardiac muscle, we're able to make a little bit more ATP per glucose molecule. So in cardiac muscle, with 1 molecule of glucose from the blood, we make 32 ATP. With 1 molecule of glucose from the blood in skeletal muscle, how many do we make?
2 less.
2 less, it'd be 30 ATP. And that has to do with the shuttle that gets hydrogen from the cytoplasm into the mitochondria. So in skeletal muscle, the shuttle is different. In skeletal muscle, this NADH plus a hydrogen ion passes those hydrogens on to FADH squared. Now, from our discussion last week, how many ATP do we ultimately get from 1 NADH plus a hydrogen ion? How many do we ultimately get? What was the figure? I'll go a little further here, because it's right here. Okay, so 2.5 ATP is the ultimately that's what we get per NADH plus a hydrogen ion. Whereas we only get 1.5 ATP per FADH squared. Okay, so in cardiac muscle, these hydrogens from NADH plus a hydrogen ion are passed across the mitochondrial membrane using the malate aspartate shuttle. These hydrogens are simply passed on to another NAD. So we're still getting 2.5 ATPs here. So if we're getting 2.5 ATPs and we're getting 2 here, okay, how many ATPs is that equal to? 2.5 times 2 is 5. Okay, skeletal muscle works a little bit differently. Okay, so these hydrogens are not passed across the mitochondrial membrane to another NAD. They're passed across the mitochondrial membrane to FAD in skeletal muscle via the glycerol phosphate shuttle. So because FAD ultimately results in 1.5 ATP, if we're talking 5 ATP from 2 NADH plus a hydrogen ion versus 2 FADH squared, how many ATP do we get per FADH squared then? If there's 2 of them, 1.5 times 2 is 3. So we've got 5 if it's cardiac muscle. We've got 3 if it's skeletal muscle. So when you're looking at the total balance sheet, if it's 1 molecule of blood glucose in cardiac muscle, the total ATP is 32. If it's 1 molecule of blood glucose in skeletal muscle, the total ATP is what? It's 2 less. It's 30 because of this difference. And that's the only difference. So it has to do with how do we get the 2 NADH plus a hydrogen ion produced in glycolysis. How do we get these hydrogens and associated electrons into the mitochondria? If it's cardiac muscle, it's no problem, because it's just another NAD waiting on the other side of the membrane in the mitochondria that picks them up. Skeletal muscle is different. So FAD takes these hydrogens and associated electrons across the membrane when it's skeletal muscle. So that there's a net difference of 2 ATP because of this difference. Okay, does everybody understand cardiac versus skeletal muscle? That's the only difference. So we're gonna go through everything and add them up and where all of the ATP comes from. So how do we get to 30 or 32 to begin with? We're gonna go through that. Okay, so G words. Glycogenolysis is breakdown of glycogen. Glycolysis is a 10 or 11 step process. 10 or 11 step process. What determines whether it's 10 or whether it's 11? What's the question? Whether we have sufficient what? Oxygen. If we have sufficient oxygen, it's 10 reactions. Glucose to pyruvate. Plenty of oxygen. Where's the pyruvate go? Goes into the mitochondria, right? Okay. What if we don't have sufficient oxygen? What if the exercise is very intense? What if it's the 400 meters or the 800 meters where we can't just rely on oxidative metabolism? We have to cross that anaerobic threshold into the glycolytic system. We sometimes call that fast glycolysis. Insufficient oxygen to meet the energy demand. So now it's an 11-step process. Pyruvate is converted into what? Lactate, right? That's the 11th step. Lactate. Is lactate a waste product? No, it's not. Because we can use lactate. So during the recovery process, we take all of that lactate and we shuttle it to the heart muscle and we shuttle it to other active skeletal muscles and we convert it back to pyruvate. And during the recovery process, now we have plenty of oxygen, so that pyruvate is just going to go into the mitochondria. Everything proceeds as normal. Now, what is the difference between slow glycolysis versus fast glycolysis? Fast glycolysis is what we call the glycolytic system. It's anaerobic. We don't have sufficient oxygen. Here's another key difference. Fast glycolysis uses glucose from intramuscular glycogen. So do you remember with glycolysis, we can get glucose from one of two sources. We can get glucose from the blood, or we can get glucose from glycogen stores, right? If it's fast glycolysis, glucose is going to tend to come from intramuscular glycogen. If it's slow glycolysis, so plenty of oxygen, for slow glycolysis, glucose comes from the blood. That's an important difference. Why is it important? What is the net ATP from glycolysis if the glucose is coming from glycogen? It's 3. So when the glucose comes from glycogen, how many ATP do we have to invest to get the whole process going? We just have to invest 1. Let's go a little bit further. Gluconeogenesis, creation of glucose from a non-carbohydrate source. Let's look up here because this is the question. So the first 5 reactions we call the energy investment phase. So we're spending ATP. We're investing ATP. And so if we're getting glucose from glycogen, we're spending 1 less ATP. If we're getting glucose from the blood, we spend 2. So reactions 6 through 10 take place twice. So you can see out here 2 times, all the way down. And so we end up making 4. And so our net, if glucose coming from the blood, slow glycolysis, 4 minus 2 is 2. If it's fast glycolysis, glucose coming from glycogen stores, okay, 4 minus the 1 is 3. So we've partially broken down glucose at this point. 10 reactions to pyruvate. Plenty of oxygen, pyruvate goes into the mitochondria. Insufficient oxygen, pyruvate is temporarily converted to lactate. Okay, so questions on this before we go further. You understand fast glycolysis versus slow glycolysis. Can you remember that we get glucose from glycogen when it's fast glycolysis? We get glucose from the blood when it's slow glycolysis, and that's important because we end up with one more ATP. Okay, let's talk about a couple of other things before we move from this slide. Key points, very, very important. Rate-limiting enzyme of glycolysis, phosphofructokinase. So reaction number 3. Why is it the rate-limiting reaction? What's the deal with a rate-limiting reaction? Yeah, it's the slowest, so it's the bottleneck. So we have rate-limiting reactions at each stage, except stage 2. So in the Krebs cycle, we have a rate-limiting enzyme that we'll get to, and then in the electron transport chain, we have another rate-limiting enzyme. So the reaction can only proceed as fast as the rate-limiting enzyme can work, right? Because that holds up the whole process. So with training, we get more rate-limiting enzymes. We get higher activity of rate-limiting enzymes so that we can better provide ATP for exercise. The other key point, ATP from glycolysis is produced through substrate-level phosphorylation. What does that word phosphorylation mean? So let's break this down. To phosphorylate something, what does that mean? What does it sound like it means? To take a substrate and add what to it?
Oxygen.
A phosphate. Okay? To take a substrate and add a phosphate. So essentially what we're doing is taking ADP and we're doing what? We're adding a phosphate, which then makes it what? ATP. That's what substrate level phosphorylation means. Where do we get the phosphate? We get it from these substrates. So biphosphoglycerate, okay? That's where the phosphate comes from. So it's different than the electron transport chain, as we'll see. So we have a few sites where ATP is made through substrate level phosphorylation. We're taking a substrate, taking a phosphate from a substrate and giving it to ATP, or ADP, so that we've made ATP. Does that make sense? Everybody? Okay. Okay, so a little bit about enzymes here. Enzymes allow for reactions to take place at a rate that sustains life. Okay, so it doesn't necessarily make a reaction happen, because a reaction would happen anyway, but it just allows reactions to take place fast enough so that the ATP we get is sustainable for life and other activities. Okay, so I'm not going to go over this slide too much. This is unless you have specific questions. So I want to get on to stage 2, 3, and 4. Okay, so stage 2, formation of acetyl-CoA. So a couple things. We are starting to make carbon dioxide. What do we do with carbon dioxide? Yeah, we exhale it. So it's made in the mitochondria, so eventually it has to make its way to the blood, and we have a way of carrying it. I don't want to go into detail on that in this chapter, but eventually it makes its way to the lungs. We exhale it. Talk about that later. We're also making another NADH plus a hydrogen ion. Okay, so at this point, where are we? Where does stage 2 take place? So at this point, we're in the mitochondria. Okay, so at this point, we have sufficient oxygen. Pyruvate is in the mitochondria and is converted to acetyl coenzyme A. So pyruvate has 3 carbons, acetyl coenzyme A has 2. So we've taken some of the carbon away with production of carbon dioxide. Okay, have we made any ATP in stage 2? Nope. Nope. No additional ATP at this point. Okay, so stage 3, Krebs cycle, sometimes called the citric acid cycle, so 8 steps. And so the rate-limiting enzyme of the Krebs cycle, isocitrate dehydrogenase. So a good test question would be, what are the rate-limiting enzymes of glycolysis, of the Krebs cycle, and of the electron transport chain? So those are 3 enzymes that you need to memorize. So for this one, isocitrate dehydrogenase. So that's important, rate-limiting enzymes. That's what holds up the entire cycle. So the cycle doesn't function any faster than what the rate-limiting enzyme can do. So what else are we making at this point? Do we have any ATP production through substrate level phosphorylation? Take a look at the cycle. Is there any point where we are rebuilding ATP from a donated phosphate? Yes, so right here, step 5. Okay, so ADP, we get a phosphate from one of these intermediates, and now we have ATP again. So that's substrate level phosphorylation. So how many molecules of acetyl-CoA do we get from pyruvate? 2. So the Krebs cycle goes around once for each molecule of acetyl-CoA. So if we have 2 molecules of acetyl-CoA, how many ATP do we make through substrate level phosphorylation? When we're all done, we make 1 per acetyl-CoA. Okay, so up here 2 times, because we have 2 acetyl-CoAs, so we have 2 ATP that are made through substrate level phosphorylation. So what else are we doing? We have NAD. Okay, and how many NAD do we produce per round of the Krebs cycle? Can you count them? Okay, so we've got here at step 3, again at step 4, again at step 8. And what else? Here, FAD is coming into play. Here. Okay, so per round of the Krebs cycle, we've made 3 NADH plus a hydrogen ion and 1 FADH squared. So if the Krebs cycle goes around once for each acetyl-CoA and we have 2 acetyl-CoAs, once the Krebs cycle is completely done, how many total NADH plus a hydrogen ion? We have 6. How many FADH squared? 2. So let's think about this for a minute. We're going to add this all up, but just think about this for a second. So per acetyl-CoA, how many ATP do we produce? For 1 acetyl-CoA, how many ATP do we ultimately produce? So 1 acetyl-CoA, we get 1 ATP through substrate level phosphorylation. We make 3 NADH plus a hydrogen ion. How many ATP do we get per NADH plus a hydrogen ion? 2.5. So what's 2.5 times 3? 7.5. 7.5 plus, how many do we get through FADH squared? 1.5. What's 7.5 plus 1.5? Is 9 plus how many through substrate phosphorylation? 1. So how many total do we get per round of the Krebs cycle? 10. So that's per molecule of acetyl-CoA we get. Ultimately, we have 10 ATP. So if we have 2 acetyl-CoAs, how many ATP then? 10 times 2 is 20. So we're starting to inch closer to 30. You see that? Because per molecule of glucose, if we start from blood glucose, what's our net? It's 30, right? So we're up to 20. Can we add in what we get through? Maybe let's wait. Let's wait till we get everything together before we go there. So okay, so everybody understand what's going on here. Okay. I'm thinking, why don't we just go ahead at this point and just add this up. Okay, so take out a sheet of paper. And I will also post this on D2L, but I want you to have this in your notes. So we're going to go net ATP needs to equal 30. So we'll assume it's glucose from the blood. So glycolysis. Okay, so our net, if it's glucose from the blood, is 2. From substrate level phosphorylation, right? Okay, so what else do we get? I'm going to draw this differently, so I apologize if let's see. I can get that to go away. Nope. There we go. Okay, so there we go. So we'll say substrate level phosphorylation. We have a net of 2. Okay. And how many NADH plus a hydrogen ion do we get in glycolysis? Do you remember? It's specifically at step 6. Let me go back and show you. It's right here. So it's step 6, happens twice. So ultimately we have 2 NADH plus a hydrogen ion. But what's different about skeletal muscle? To get these hydrogens and associated electrons into the mitochondria, what happens? Not in cardiac muscle, but in skeletal muscle.
Skeletal muscle, you get 3 ATP for FAD, but cardiac spike for NAD.
Okay, so it's right here. So this is just showing glycolysis. So ignore everything from this point down. So we're not to pyruvate yet. So the key point with these is that skeletal muscle uses a different shuttle system, glycerol phosphate shuttle. So it's actually FAD that ends up carrying these hydrogens and associated electrons to the electron transport chain. in skeletal muscle. So let's take that into consideration here. So ultimately, through oxidative phosphorylation, this takes place at the electron transport chain. Okay, and what does phosphorylation mean again? It means to take a phosphate and give it to ADP to regenerate ATP, is all that means. At the electron transport chain, why do you think we call it oxidative? Because it requires what? Oxygen. And it turns out that right at the end of the electron transport chain, it's oxygen that binds to these hydrogens and electrons to make what, you think? H2O is water, okay? So oxygen is the final hydrogen ion electron acceptor at the end of the electron transport chain. So that's why we call it oxidative phosphorylation, because we're adding these hydrogens and electrons to oxygen to make water. Okay, so in glycolysis specifically, the NADH plus a hydrogen ion. Okay, so we have 2 from glycolysis, right? Step 6 takes place twice. And we're giving those hydrogens and associated electrons to FAD at the mitochondrial membrane. So FADH squared. So if we're looking at the ATP that comes from glycolysis, turn that into a D, I'm sorry. How many total ATP do we get? We get 1.5 per FAD. So if we have 2, how many are we getting? 3. So we get 2 through substrate level, 3 ultimately at the electron transport chain. But they come from what happens in glycolysis. Okay, so now we're going down to the Krebs cycle. Okay, so substrate level phosphorylation, because we have 2 acetyl-CoAs, okay, and each acetyl-CoA is broken down through 1 cycle. How many ATP do we get ultimately through Krebs cycle substrate phosphorylation? 2. Yeah, remember step 5, so that's, we'll put a 2 here. Okay. All right, so oxidative phosphorylation now through the transfer of hydrogen ions to the electron transport chain. Okay, so we've got a total of 6 NADH plus a hydrogen ion. And we've got a total of 2 FADH squared. Okay, so how many do we get with 6? What's 6 times 2.5? Be 16, right? Okay, so we get 16 there at the electron transport chain. 2 FADH squared. 2 times 1.5 is 3, right? So 2 plus 3 is 5, plus 2 is 7, plus 16 is 23. Okay, plus 3 is 27. So where's the other 3? What are we missing? I want you to see if you can look at it and figure it out. So I want you to discuss, and how do we get to 30? Where are we missing ATP? So I want you to just take a minute, look through your notes, look through the PowerPoint, see if you can figure out where we are missing that extra 3. So we have 2, there's 2 there, 3 is there.
So in the glycolysis, in the oxidative phosphorylation, I have a question.
Sure.
We're only doing one or the other for NADH or FAD because it's based on the skeletal muscle.
Skeletal muscle versus cardiac
and then
muscle.
once you're in the mitochondria, doesn't matter. You do both.
In the mitochondria, you're already in the mitochondria.
So
You would just do both, yeah. Take a minute, take just a second, look it over. So we're up to 27. Where do we get that additional 3 from? How do we make that 30? Right down here, take a look at. Take a look at this slide right here, slide 22. So this kind of is nice because you're able to compare cardiac muscle, heart muscle versus skeletal muscle. So notice in stage 1, if it's from glucose versus skeletal muscle, you're getting 2 less because of the difference in hydrogens across the mitochondrial membrane. So I want you to take just a second and think about it. So here's that transfer in skeletal muscle, remember? Okay, so rather than 2 NADH plus a hydrogen ion, which would be 5, we're getting 2 FADH squared, which is 1.5 for that. So it's only 3 ATP versus 5 ATP. So that's why that part is highlighted. Okay, take a second. This is important to understand where everything comes from. So what are we missing?
The 5 from NADH
Everybody see that? Stage 2 is what we were missing. So stage 2 is what?
Formation of acetyl-CoA.
Yeah, formation of acetyl-CoA. We make NADH plus a hydrogen ion, which 2.5 per molecule gives us an additional 5 ATP. So I really want you to look this over and kind of get set in your mind where all of those ATP come from. Going from glycolysis to the electrons transport chain, oxidative phosphorylation, stages 1, 2, and 3. Okay, so I think at this point, I think it would be good to kind of get some group activities going. So I would like to start on this today, and we can continue as we review for the exam on Thursday, but I'd like you to get in and review some of this stuff from the chapter. And this is like we did with chapter 1 with the review activities.
Thank you.
Yeah. Okay, so remember on chapter 1 activities, if the true or false, if it's false, what do you need to do? Yeah, you got to write in what the correct is. So let's do the first one together. So the first law of thermodynamics states that energy can either be created or destroyed, but not changed in form. Look at the wording.
False.
It's false. Why is it false?
Energy can neither be created nor destroyed.
Neither be created or destroyed, not either. So the wording there would make it false. Neither be created or destroyed, but not changed in form. Let's do another one. So, question number 2. ATP is constantly being hydrolyzed and resynthesized. The amount in skeletal muscle at rest can supply about 3 minutes of maximal contraction. So we do have a small amount of stored ATP that lasts us for about 3 seconds. After that, we need our metabolic systems to meet the energy demand. So that would be false. So you would write, we have enough to last for 3 seconds of maximal contraction. So we have a little bit of storage.
So it would be the difference for cardiac muscle. So it's skeletal muscle. Does that matter?
For number 3?
No,
Or-
for number 2, it's just talking about skeletal muscle, so it's asking what the difference is.
I'm not sure how much cardiac muscle has in storage. I would assume some, because it has to be, I mean, that's operating from birth till death. So there'd have to be some there. But that's a good question because it ties into number 3. Brain cells cannot produce energy anaerobically, and cardiac muscle cells have only a minimal capacity for anaerobic energy production. So that was a little bit early on in the PowerPoint, but that is true. That is true. So skeletal muscle has a much greater capacity for anaerobic energy production versus cardiac muscle and especially brain cells. Yeah, let's keep going. So pyruvic acid, pyruvate, is the universal or common intermediate in the metabolism of carbohydrate, fat, and protein. That's false. So what is the common intermediate? Acetyl-CoA. Very good. Okay, so why don't you work together on this? See how far you can get today. So I wrote up at the top right the parts that are most important. So these are the parts that I want you to complete, the parts that I would include test questions from. So you can see, top right, exercise 1, exercise 2, exercise 4, parts B and E. So just 2 parts on exercise 4. If you want to turn over the page to exercise 2, I can help get you started on exercise 2. So what we're looking at is metabolism of glucose, protein, carbohydrate, and fat. So under protein, question number 1, you would put polypeptides. So a polypeptide is simply a bunch of amino acids that are bound together and shaped into a protein. So number 2, if you break down a polypeptide, what do you get? Yeah, amino acids. So I want you to go down to number 10 on exercise 2, and you notice that's where the protein, carbohydrate, and fat all come together. So what would be the best response there for number 10? Yeah, so that's where acetyl-CoA would come into play. So I'll let everybody get the idea on what to do for number 2. So I'll let you fill in the rest. Exercise 4, part B. This one might be best after lecture on Thursday, because we're going to go in and talk about the electron transport chain and the steps for that. But that's basically what you're doing, is outlining steps for the electron transport chain. So I encourage you to go into your textbook and look that over, even do the activity, but that's going to be lecture for Thursday's electron transport chain. And then exercise E. Exercise E, you're just looking at the different reactions and matching. So this one also would be good to try after the lecture part on Thursday. So we need to talk about words like polysis and lipogenesis and beta oxidation. Those are things that are going to come up on Thursday's lecture. All right, so go ahead and take off and work together, and I can come around and help you with ones that you may get stuck on. like a number 5 there. They flipped around the definitions. Number 5.
Good. I'm just thinking through number 6. I'm like,
It's not trapped into the muscle cell, is it? Can't it be released into?
Now, once in the muscle, it won't leave. So that is
Okay.
true.
Okay.
then
So the
we-
liver is different. Liver's maintains blood glucose. But
So if we see any kind of blood glucose, we know it came from the liver. So it never comes from the muscle.
yeah, so muscle will hang on to it once it's in there. What's the rate limiting enzyme for glycolysis again? Give you a clue on number 7. Yes. Yeah, very good. Everybody get true on number 8? So we talked about number 9 today.
Right?
Wait a minute. So slow glycolysis, where do we get our? Yes.
So that would be lactate.
Slow glycolysis is glucose from the blood. Fast glycolysis is glucose from muscle glycogen. So part of it is right. So for slow glycolysis, the end product is pyruvate, but it's where the glucose is coming from that is incorrect. The primary fuel for slow glycolysis is not glycogen, it's glucose from the blood. Whereas for fast glycolysis, you're getting glucose from glycogen.
So they just have the fuel switch.
Yep. So you could just write the fuels are switched and put false. Yep, so keto acids are not the same thing as ketone bodies. That's different. So pyruvate, acetyl-CoA, all those 8 intermediates in the Krebs cycle, those are keto acids, which is different than ketones or ketone bodies. are associated with a ketogenic diet. You should be able to get number 11's.
Can you kind of just talk me through number 12, like what it's saying?
Number 12.
My brain's like,
Yes, so remember substrate level phosphorylation. So up here we have 2, where you have a phosphate donor to ADP to make ATP.
right.
And then down here in the Krebs cycle we have 2. Down here in the Krebs cycle we have 2 more.
Okay.
Whereas in the electron transport chain, that's where we're passing those hydrogens and electrons that we'll cover on Thursday. So that's oxidative phosphorylation, because you have to have oxygen.
Okay.
So number 12 would be true. Number 13 we'll get to on Thursday, but that's the other rate limiting enzyme. 13 is true. When we get to electron transport chain on Thursday, cytochrome oxidase. So there's 3 rate limiting enzymes that you'll need to know for the exam. Okay, so that's another one from lecture. So Jackie asked a good question. We haven't quite got to that yet in lecture, but all of the triglycerides are turned over every 3 to 4 weeks. So that would be number 15 is false. So that's something for Thursday, but it's every 3 to 4 weeks is the answer on that one. Number 16 is also a Thursday topic. So rather than triglyceride, it's glycogen. So 2.5 grams of water are stored in muscle per gram of glycogen, not triglyceride, glycogen. Report 17. It's true.
Okay. I'm like, I was
That's
thinking it's true,
another
but I'm like,
one for Thursday. It's
What
called
was that
beta
one?
oxidation. That's oxidative deamination. So it's beta oxidation of fatty acids for question 17. Beta oxidation. Yes, so you want to look up what the branched chain amino acids are on that one. So you want to, it's not alanine, glutamine, glutamate. So for 18, you'll need to look up what the branched chain amino acids consist of. What are the 3? So if we do use amino acids for ATP, the branched chain amino acids are commonly used, but it's not the 3 that are listed there.
Like, look at a line.
3 branched chain amino acids. or at the end of the textbook chapter would have them as well. Yes, ma'am. So Ashley's got number 19 is true. Has anyone looked up what those BCAAs are?
Leucine, isoleucine, valine.
Mm-hmm. Perfect. So, extended question, what would, if we are converting an amino acid to glucose, what is the G word for that? Gluconeogenesis, right. So,
What were they leucine, what?
Isoleucine,
Yeah.
valine.
20. That is the bottleneck, yep. So pathway only operates as fast as the rate limiting enzyme. Yes, that would be true. So the rate limiting enzyme is the one that determines how fast. So like I said, this test won't include the hormones and amino acid stuff. So 21 is true. Growth hormone and cortisol both promote fat mobilization and amino acid uptake. Yes.
22 is true, right? Or is it?
Yeah, no. Let me just give you a clue on that one. So this slide right here should help you with number 22. Right at the end of the PowerPoint. So that's why a marathon athlete or ultra endurance athlete would undergo carbohydrate loading because they could increase this value. So glycogen is a polysaccharide. So glycogen is that's the polysaccharide. So on number 2, you'd want to start a carbohydrate with polysaccharide. Take a look here. So. I'm still thinking. And Jeff for 15, so you put these two, and then beta oxidation is correct. So once you've gone all the way through exercise number 2, I think that'd probably be a good stopping point for today, because the other activities will be with Thursday's lecture with the rest of chapter 2. Don't forget, what's the other part of triglycerides? Another one of those G words, yeah. That would have to go probably here, and free fatty acids here, and beta oxidation here. Are you okay? Good. Shay, feeling okay? Finishing it up?
I think so.
Yeah, then don't forget about, yeah,
Okay.
so it's in there. Usually we take the glycerol and we just make glucose out of it.
Okay.
Are you finished? Michael, those things.
Finished it up, let's do that.
That is. That looks good. Not at all? Good. So you'll turn in this whole thing on Thursday. So yeah, we'll do,
We'll finish that.
yeah,